First of all, thank you everyone for joining us. Good afternoon, good morning to those of you past to the west of the Eastern time zone. My name is David Regan. I'm Chief Executive Officer of Sona Nanotech, and I'm very pleased to have you join us today and welcome you to Sona Nanotech's first investor webinar of 2024. It's been a couple months. We've got some updates we're very keen to share with you. I would emphasize the intent of this particular webinar is an update on progress and to share some new perspectives on our path forward. If you're looking for a more fundamental understanding of Sona's gold nanorod technology and our current study at Dalhousie University, please consult our website, which has a number of webinars and white papers on those topics. Also, there should be a chat button at the bottom of your screen, somewhere on your screen in Zoom, that permits you to send us questions, which we will look forward to addressing at likely at the conclusion of the presentation. Now, before we get started, we note that we'll be making a number of forward-looking statements in this discussion, so we'd refer you to the company's disclosure documents on various risks inherent in an investment, which are available at sedar.com. Now, we have 5 items to go over today, and joining me on the call today, I have Sona's Chief Scientific Officer, Len Pagliaro, PhD, as well as a special guest in Dr. Carman Giacomantonio, who is the principal investigator of the study we have underway currently at Dalhousie University. So we'll hear a bit of an update from Carman shortly here. Now, just want to provide very brief background, given that that the purpose of this webinar is for an update. But it's important to understand, what are nanoparticles? Nanoparticles are, in fact, highly useful because they have unique optoelectronic properties that allow our scientists to manipulate them to serve many useful functions. Now, what Sona has, many people make nanoparticles, many people make nanorods, gold nanorods. What Sona has is we specialize in the one particular type of nanoparticle, a gold nanorod, that provides perhaps the greatest degree of functionality and certainly the greatest efficiency in converting non-thermal energy into heat. This is highly relevant for our particular targeted hyperthermia therapy. Sona's advantage is that the patented and patent-pending gold nanorods we have are the only ones that are made without the use of toxins, making them ideal for the use in advanced nanomedical applications in vivo. Now, what are we doing with this advantage? We're leveraging it right now by developing a photothermal cancer therapy that aims to destroy cancer tumors from the inside out without the cutting, burning, and poisoning of patients that currently happens with most cancer therapies. Now, I'm going to kick it over to Len to give us a little bit more background on Sona's gold nanorods and what makes them unique and useful. Len.
Thanks, David. So there are 2 things that are really valuable about Sona's nanorods. The first is that they're uniquely biocompatible. They have been vetted extensively by a program called the Nanotechnology Characterization Laboratory. It's a national lab in the US funded by the FDA and the National Cancer Institute. And they are sterile, they're free of endotoxins. They've been highly, highly vetted, very uniform particle distribution, as you can see in the image here. And so the ability to manufacture these nanorods without the toxins, as David mentioned, is something that Sona really has uniquely. Now, the FDA has allowed gold nanoparticles to be injected into humans in clinical studies, but none has been cleared yet commercially. So none is a commercial product on the market. So it's still relatively early days for these nanoparticles in cancer treatment, but they are in humans. And so that's a good thing from our perspective, from a regulatory angle. The NCL, as I mentioned, has done these studies, and it's kind of like having a good housekeeping seal of approval in the nanotechnology world. Very important. It gives a lot of credibility to these materials. And so on the next slide, we'll see the second thing that's really unique, and again, David alluded to this, is the ability to generate hyperthermia, the ability to generate heat. And so David mentioned photothermal therapy, converting light into heat. So converting non-thermal energy into thermal energy, basically. The nanorods do this extremely efficiently because of this characteristic called surface plasmon resonance. They can convert that infrared light into heat, and they do it very, very efficiently. Now, the heat is important for cancer because, as this diagram shows, there's a range of temperatures which are above body temperature that do very important things to tissues. And importantly, cancer cells are more sensitive to heat than non-cancerous cells. So our target temperature is in the range between about 44 and 48 Celsius. By getting into that temperature range, you can see that there's inactivation of cells, again, specifically cancer cells, because they're more heat sensitive. And then you can get up to a stage of necrosis, cell necrosis. That's an important temperature range because it damages cancer tissue much more than it damages healthy tissue. Healthy tissue can withstand it.
That's great. Thanks, Len. Those are key advantages that are important. So looking at the current focus of the company and where we're going in the future, to set some of the context here before we get into comments from Carman, it's important to note that what we're doing is leveraging the uniquely biocompatible nature of our gold nanorods to develop what is effectively a low toxicity, high efficacy, gentle cancer therapy that are powerful but gentle, and that will involve, as we say, neither the burning, poisoning, or cutting that are seen with many different treatments for patients for cancer care. The other focus, obviously, is the study to validate Sona's technology for melanoma and breast and colorectal cancer in mouse models, and together looking at them and the effect they might have with current leading immunotherapy, immunology therapies, that is. Next, we're going to talk about what the results from the study could mean for us. And looking forward, whatever the results of the innovative study, they're going to help us. The results of this study will be instrumental in helping us guide the direction for new areas of research, potential new future offerings, and of course, what we expect the data will support submissions to regulators as we push to get this into clinic, secure regulatory approvals, and eventually commercialize it. I would anticipate that at least the last of these components would be done with an established partner, if not the first step, based on how the results of our study look. Our goal for the intermediate term is to have our uniquely biocompatible gold nanorods treating cancer in humans. Once we do that, in the longer term, we would anticipate Sona's gold nanorods becoming the standard nanoparticle for cancer treatments, rheumatology, or genetic disorders, or some new applications worldwide, based on the credibility created through this and future studies. Now, I want to welcome Dr. Carman Giacomantonio, our principal investigator on the study, to share a few more thoughts here. Carman, you're sideways and you're labeled as David Regan, but that's fine. There we go.
There you go.
My twin brother clone. If only it were so. Here's Carman.
Our parents are not very happy with this designation, so well, at least yours. So thanks, David. So and Len. So essentially, what we are hearing is that this gold nanoparticle is being used initially as essentially a tumor destruction agent or a primary tumor treatment agent. And what we're branching into with this discussion is it as an adjunct to an immunotherapy, or even an adjuvant to immunotherapy. Next slide. Yeah. So, as Len mentioned, we know that cancer cells are particularly sensitive to thermal energy. Cancer cells thermal energy essentially drives the cancer cell into a death cycle that normal cells are generally resilient to within a range of temperature. One of the things that's particularly important about the gold nanoparticle is we can control the amount of energy that's generated. What happens is, when we inject the particle either intravenously or directly into the tumor, we can activate it specifically where it lies. The targeting is within centimeters of the energy source and limited to activation just of the particles that experience the energy from the near infrared light. This is very important because many of the other targeted therapies that we currently struggle with in health have more global effects, either systemic immunotherapy, systemic chemotherapy, or radiation. And radiation has, as many of you would know, a collateral damage challenge that we're left with dealing with the aftermath, both in the either the surgical setting or in the healing setting. So immunotherapy, we know, is currently leading all cancer therapies, and it's been superbly effective in some cancers, yet in other cancers it's had limited efficacy. And there's lots of theory as to why it would have an ability to treat a melanoma quite effectively, but not treat a breast cancer effectively. Probably the most primary reason that this doesn't happen is the antigens that are generated in these cancers, the signals that excite or interest your immune system are just not apparent. The gold nanoparticle promises to enhance that by creating local tumor destruction, and with local tumor destruction, as a part of the process of cleaning up the normal biological process of cleaning up the tumor necrosis, the dead tumor, is the presentation of some of that tumor to the immune system. We have seen this in other kinds of cancer therapy, radiation therapy, some toxic therapies, that when that happens, serendipitously, the immune system actually recognizes the cancer in a new way and takes over and kills the tumor. This is the strategy that we're focused on here. Next slide. So what happens? So we're in the early phases, and what will success bring us from the lab? So the first thing is that it'll confirm that we can kill the tumors, we can shrink the tumors, we can cause the necrosis that we've been talking about in a controlled fashion in the mouse model. We're going to try this in breast cancer, melanoma, and colon cancer. The second thing is we're hoping that we can enhance the immune experience. And so what this means is this: we anticipate, with good evidence to support this hypothesis, that the thermal destruction of the tumor will generate the antigen that we spoke of. And this, in combination with some local immune therapies, should create the immune-directed response that we believe is at the core of this tumor killing. So what I'm saying is that although the thermal energy will drive the cell into a death cycle, the actual clearance of the tumor is almost assuredly an innate immune response. So the final thing is, next portion, is that there is a reasonable anticipation that at least in some of these animals and some of these experiments, we'll see a response that goes beyond the primary tumor. Why do we expect that? Because tumors, as you know, are associated with not only local growth, but developing of metastasis. Those metastases, or daughter cells, or offspring of the tumor, will very often look exactly like the primary tumor. So when you generate an immune response to that primary tumor, given that the immune response is truly a systemic response, in that the cells that are released have to circulate and come back to the tumor that's been treated, any cell expressing a tumor antigen like the one that's been primarily responsible for the response would be attacked by the immune system. This is what we call an abscopal response. Now, this is a hopeful, this is a hopeful response at this point. We do see this in clinic from time to time in the context of radiation therapy. We believe our hypothesis is correct, is that we're going to see this in the animals, and we've designed the experiments to test this hypothesis. So what are the implications for all of this? What are the things? Well, the first thing is, we get the experiments done, we'll prove the primary hypothesis is that we can kill tumors locally, we can generate some kind of immunity, and and hopefully that we can reproduce. This is a reproducible phenomenon. In the context of the science world, this will allow us to present this data at local, national, international meetings. And often at these meetings, and almost invariably at these meetings, is where the potential partnerships would be struck to enable us to talk to other scientists, to look at partnerships in other labs, partnerships with industry who are constantly combing these scientific meetings to see where the next where their next opportunities are, to not only to learn new things, but to add on to the strategies that they're already using. And this is clearly where this will ultimately go for us. Next slide. So the enhanced immune response that I'm talking about, that I'm really excited about in terms of this work, is demonstrating that the immune pathway that we've established is, or the killing pathway we established, is immune-based. And then presenting the strong evidence from our research to other labs that may want to collaborate, and to other companies that may want to support or even enhance what we're doing, showing them that the scientific pathways, the biological pathways that we have established, are reproducible, they're understandable, they're not serendipitous, but they're intentional.
And that's been the challenge to date, right? I mean, it shows up occasionally, but it's a question of making it.
Well, this is the big challenge with abscopal responses, for sure, is they're inconsistent. We see them, and we know that these responses happen, and for the most part, many of them are phenomenal. Even to this date, when we use current immunotherapies, we know the immunotherapy is working because it's enabling the immune system to continue to do what the immune system does. But we know with current therapies that without an established immune relationship, the immunotherapy that we give won't work. It won't enhance what doesn't exist. The therapy that we're talking about with the immunity initiated through tumor destruction from the thermal energy of the nanoparticle is new in immunity. And so in the context of the harmony of doing this with the immune system, our hypothesis is that we're going to generate new antigen that's tumor specific, that is systemic. That's the abscopal response. We need to take it from phenomenon to process.
That's fair. Would you say, we've got a question here, but asking if whether the combination of targeted hyperthermia therapy, our tumor destruction therapy, together with an immunotherapy, would result in an easier path through clinical trials and approvals?
Well, that's a great question, and the short answer is it's a logically easier path, because we have current immunotherapies that have already been rigorously tested in the clinical setting, and so the safety and efficacy is understood, and so are the limitations. And so clearly, if we combine that with the gold nanoparticle, which we anticipate will be very well tolerated in the clinical setting with minimal toxicity, this is the natural next step, and it's the easier path, because again, if my contention is correct that the gold nanoparticle resulting tumor destruction is ultimately an immunological pathway, then certainly the checkpoint inhibitors will advantage the immunology, because if the antigen that's exposed is yet again a weaker antigen, using the checkpoint to address that concern should enhance the immunity. That's the right hypothesis, and it is the natural pathway for this to move, as the kind of low-hanging fruit, if you will. The finesse, the elegant science beyond this will be combining the two within the tumors to more critically analyze the tumor microenvironment, the antigens, the T-cell receptors, et cetera. That will be a part of the tomorrow's research.
Excellent. Thank you for that, Carmen. I don't know if there's anything more, but if you could stick around for our Q&A, we'll have that in a few more minutes. And,
sure.
and certainly it is our expectation that, yes, as you put it well, it's a logical path forward to see existing products in market looking to have enhancements to those products and extensions to their life by adding on capabilities such as the ones we're exploring right now.
Exactly.
So as far as recent developments go, we have three to share with you in general. Now, first, after a disciplined selection process, a lot of work has gone into this. We've identified several partners with whom to work to provide the GMP manufacturing services, the safety and toxicity studies that will be required by regulators as well, pardon me. Two of these entities are based in the US and one is in Europe. And it's really important to understand that selecting the right partner is critical because they've got to have the right capabilities for what we're going to need now and also probably in a year and 2 years' time at the right price and at the right size. We don't want to get lost in the bureaucracy of a large entity that's so large that we're meaningless to them. We need folks, and we have vetted these parties to be both capable and engaged and entrepreneurial, frankly. And I think we've done a great job on that, and those partnerships are now primed and ready to go. And those are key supplier relationships, obviously. Secondarily, IP protection is critical for the business and critical for the business to capture the economic benefits of the good we expect to create with targeted hyperthermia. We've been very active with counsel on building an IP ring fence around the business as a result. Well, our gold nanorod patent has been granted in one territory. It is still pending in others, but we expect more approvals soon based on the back and forth we've had with investigators. We are also in the midst of filing several other patents, including for our next-gen light device that we've spoken of in the past and will be important in terms of operationalizing targeted hyperthermia therapy in the future. There are other exciting concepts, though, that we're working on, and we'll look forward to be able to tell you about those in the future in the coming months. Now, finally, as far as updates go here, with the Excite International ETR panel insights, we have labeled ETR as Early Technology Review. I'm pleased to report that we held the first of our several 3-hour panel discussions with a group of surgical oncologists and executives from U.S. payer organizations as part of our partnership with Excite International. As some of you may know, Excite, which is a not-for-profit global collaborative initiative, facilitates feedback from experienced practitioners and payers for emerging medical device developers. The purpose of this first session was to introduce the group to our proposed therapy and to have them challenge us and brainstorm with us on the most efficient route to clearance by regulators, to market, and ultimately to adoption by their fellow practitioners. Getting this input up front is critical, as there are plenty of FDA-cleared medical devices sitting on shelves out there that have never succeeded in gaining adoption for a plethora of reasons. Now, while I can't name the individuals on the panel, I did want to give folks a sense of the credibility they bring, and so I here cite the institutions with which they're affiliated. Needless to say, these folks are highly credible, and they were highly engaged in the process. Whether they currently work for a medical school, hospital, or payer organization, all of them are physicians with broad, relevant experience. The good news is, coming out of that first very engaging and interesting session, is that they were very supportive of targeted hyperthermia therapy and all saw the merit in it. The better news is that they challenged us on the best path to approval, questioning both the best indications for use and practical considerations when using an endoscope. And they brainstormed with us on possible enhancements to both. Based on the discussion and on our ongoing research, reflection, various consultations, we'll be reconvening with that group in the coming weeks with a greatly improved protocol and proposition to gain their further insights and guidance and support, to make sure that they have heard, that we have heard their commentary and their guidance to us and incorporated it to make for a stronger proposition for Sona as we move forward for targeted hyperthermia therapy. That's what we have for you today. So at this point, we'd be happy to take further questions along the line. Let me check again. There's a chat bar there. And I've got a question here. Are there any LOIs in place with said suppliers yet? What level of commitment can they give? We have LOIs available to us right now. These are basically draft contracts that are being negotiated. Again, we've vetted each of these parties, and we're at the point now where we're ready and can execute those shortly when appropriately. And what level of commitment can you give us? What level of commitment? Well, commitment, certainly we have those suppliers are giving us a high degree of commitment. We've vetted that as part of the process in terms of whether they have the availability in their labs and whatnot to help us. And so we're really, really feeling very good about that. Do have a question in here, a few coming through. Pardon me. So there's a question here, which is fair enough. Have you met with the FDA? Well, I'm pleased to report that we've been back and forth with the FDA, and we have a meeting pending with them coming up next month. We're excited about that. It's a long lead time in order to get a meeting with them. And the process is interesting because we had to put in a 50-page submission, effectively, completely outlining our proposition and the support for it. We also included with that 15 specific, concise questions for guidance from them. The FDA now has the obligation to reply to us with answers 5 days before the meeting. And with that, we'd expect they'll have follow-up questions. So it's a very organic back and forth conversation that occurs. But that's a critical one. That's one where, as we've mentioned in the past, we're fortunate that the folks from the nanotechnology characterization lab have proactively offered to join us. They'll be on the call. Dr. Giacomantonio will be on the call with us. It should be an excellent session. Let's see what else here. Are the partners taking a financial position in Sona? Well, right now we have suppliers we're working with. As Carman mentioned in his remarks, once we have study results and if published and taken to the major cancer conferences, that's where those real discussions take place with partners. And we'd be looking forward to having meaningful conversations with them about potential partnerships at that time, because that would be the appropriate time. And any terms of which, of course, would have to wait for that point. Are you looking to generate a revenue stream from the sale of gold nanorods? Len, do you want to speak to that?
Well, sure. I do think there's potential there. There have been some very modest revenues from the sale of gold nanorods. Several issues to consider here. I think one is that, of course, we don't want to compromise our own mission and the proprietary nature of our gold nanorods that specifically are for photothermal therapy. However, there are many groups who are interested in using them for other purposes, diagnostics, lateral flow assays, academic groups who are doing basic research. And so I think there's good potential for that to generate an ongoing revenue stream in the future as that part of the business grows.
Yeah, and as I think we mentioned in the remarks, with success from this study, we expect to raise the profile of Sona's gold nanorods significantly and the credibility of them as well. And so that would be a natural platform from which to continue with sales of the gold nanorods. Another question here, obviously, we're lots of questions on this front in terms of what's the status of the study, where are we, how soon, when are the results coming, et cetera. And what I can say is that the study is underway, and we, of course, are excited to be able to share results as soon as is appropriate, but we're going to be following good governance practices here, and we're not going to be providing updates along the way or anticipating timing. We will, of course, update the market as soon as we have official results on that. But the study is underway, and we're all very, very excited about it. Separately, it appears large animal studies and immunotherapy studies are both being delayed pending more feedback on how the approach of such further studies goes. Our focus right now is on the Dalhousie Giacomantonio study is the focus, and vetting and lining up our suppliers to be ready to be able to do those studies. But I would also say that they're subject to conversations with the FDA, because we have had, in one of the specific pieces of feedback from the the Excite group was, why are you planning on going to large animals? They didn't expect that the FDA would require that, given the move in recent years by the FDA to diminish any such studies where and when possible, and certainly given the low toxicity, high efficacy of what we believe we have here. So we don't want to jump into something that may not be needed. Got to make sure apples are all lined up in the right order here. Can we expect to see announcement on the completion of the infrared light device soon? Len, do you want to speak to what we're doing with the light device?
Sure. So we do have the next generation prototype model. It is in Dr. Giacomantonio's lab at Dalhousie University now, and it's been approved by their laser safety committee. The device does contain an infrared laser, and so that device is ready. Now, this is not the device that will go to market eventually, but it's the next iteration of the device that has the capability to put infrared light through a very skinny fiber, a very narrow optical fiber, which is compatible with an endoscope. For the rodent studies, we don't need to use an endoscope, but it does have the ability to deliver that infrared light and then also to monitor the temperature of the target in two ways. One with little needle thermocouples that go into the tumor tissue, and then one with a non-contact optical device that measures temperature, measures surface temperature. And so I think it's going to be a really good opportunity to test the functionality of that device in animal studies.
Mm-hmm. And I would add that as we move forward, we're looking at, much broader applications of this technology based on some of the conversations we've been having that would open up other possibilities. And of course, the light device is highly specialized for one application that would be necessary, and we think will be the significant major market opportunity. But there'll be, we believe, multiple applications and therefore possibly multiple devices needed in order to hit those. Some of which would not be would need to be as sophisticated or expensive as this one. Question here: what role will Dr. Carman's papers and presentations play? It's good questions. We haven't really spoken to that, other than to say, and we should say, that the data coming out from it will be extremely important in presenting that to regulators and other parties as we go forward. There have been past studies, and perhaps Carman, you can speak to that, the type of analysis that's done and how that might differ on this study than as opposed to on past studies that have been done in there.
Yeah. So there have been some studies and that give us confidence that the technology is going to shrink the tumors. What the important bits of data that are going to be, I think, under the microscope for our next efforts are tolerability from the animals. So we've built a lot of that into our experiments going forward. What is the toxicity? What's the tolerability? And what's the efficacy in the tumor? But the other thing is the biology. So we'll be analyzing the tumors very carefully and the animal's systemic immunity to prove the hypothesis that we've not only caused the tumors to shrink locally, but we've generated a systemic immune response, and that the process that's ultimately clearing the tumors is an immune pathway. This information will be important for us moving forward in terms of partners.
Thank you. And there's another great question here, actually. That's will Dr. Carman test tumor shrinkage first before testing immunotherapy? In terms of, is it all put together or is it separate?
Yeah, so it's actually together. We're going to test tumor shrinkage, but those tumors will be subsequently analyzed in the context of the most system to understand what the biology of the shrinkage is. There's lots of methods that tumors shrink. One is necrosis; they die, and that's a death process. But there's actually an active and intentional cell death cycle called apoptotic cell death that are two different pathways, involve lots of different cellular biology, but the ultimate cleanup, regardless of the process, is likely a common immune cell death pathway. And so all of that gets tied together. But this is a simultaneous experiment, or the results are out of one experiment, essentially.
Right, but it's fair to say that we'd have in isolation, we'd know, look, is there shrinkage of tumors without the immunotherapy element in isolation?
Well, the results will the results will help define the contributions of each of the components.
Right.
Yeah.
And there's an extensive histology and analysis done.
Yeah. Yeah.
Post.
Exactly.
Okay, good. Another question: as a cancer researcher, how significant does Dr. Carman think the results of the study will be? That's asking you to speculate, I guess, here. But
yeah, I guess in speculation, this is I'm more than excited about this. These results are extremely important in terms of proving this is proof of concept. This is the kind of evidence that we need to move quickly to whatever it is the next step to get us into the clinic. So I don't want to overreach here, but these results are essential because I know most isn't a human, but the proof of concept, the most immunity is what led all of in fact, our colon cancer model is the same model that was used to initially prove the concept that PD-1 inhibition will likely change immunotherapy in humans.
that's it. Okay, thank you. Any other questions? Happy to take them now or at any time. We're available to you at the contacts you see on this page. In closing, I have to say that we're really proud of the progress that we have made after just 1 year of since the transformative acquisition of Lens SIVA Therapeutics. And we now have a stronger team, a much stronger path forward, stronger partners, stronger IP, and we're in the midst of the first of several preclinical studies that will be needed to permit a first in-human study. The entire team right now from our board, which, as we've said before, hits well above its weight for a company of this size, right down to the scientists in the lab, are all charged up with excitement for the important work we are doing, and we really look forward to sharing everything we can when we have results with you. So in the meantime, thank you for your time, and we look forward to coming back to you soon. Thank you, Carmen, and thank you, Len, too.
Have a great day. Thanks.